Hello, everyone, and welcome to today's webcast, Introducing Simcyp Discovery. Before we begin, just a few tips to help you participate in today's event. You have joined the webcast muted. Should you have any technical issues during the webcast, please use the questions icon. You will have the opportunity to submit text questions, which you may submit at any time throughout the event. We'll have time set aside at the end for live Q&A, and today's event is being recorded. I would now like to introduce Mark Laverne, Senior Vice President for Product and Commercial Strategy, Simcyp. Mark?
Thank you so much, and welcome everyone. We are very excited to speak with you today about Simcyp Discovery, a new commercial product offered by Simcyp. So let's first talk about who we have on our team. As mentioned, I'm Mark Laverne, and with me today we have Ian Gardner, one of our senior scientists supporting both Simcyp and Simcyp Discovery, and also Hannah Jones, who is our head of PBBK consultancy. Also on our team, but perhaps not in attendance today, or will not be speaking at least, are Masood Jamie, also one of our senior scientists, Ellen Langfuss, our Chief Commercial Officer, and Himanshu Mishra, who is one of our essentially business developers. So our agenda today is as follows. We're going to provide an overview both of our company and of Simcyp and the wider organization. We are going to talk briefly about the new offering, which is Simcyp Discovery, and then we will play a brief demo of highlighting some of the key features of Simcyp Discovery. And then we'll have a brief summary regarding the new product, and a brief Q&A session, and also discussion of potential next steps. For this, Q&A can generally be focused on the feature set of the product, or logistics about how to get evaluation or purchase. And then following that, for those interested, we'll have a more detailed technical discussion of some of the methodologies underlying this new software offering. So a little bit about Certara. So basically Certara is a model-informed drug development company. We offer a number of products and services in support of that mission. We have approximately every year in the past 3 years, we've supported over 200 regulatory and payer submissions. Currently, we have about 1,600 clients globally, and over 1,100 employees worldwide. So, we are the leading model-informed drug development company in the world, and 90% or more of all novel approved drugs by the US FDA have in some way, we've had some involvement, be it through our software or our services. So Simcyp, as I mentioned, we have a broad portfolio of products and services in support of model-informed drug development, all throughout the drug development cycle, including drug development and regulatory strategy consulting, biosimulation and informatics software, regulatory writing, and also health economics outcomes research. And so, the sweet spot essentially for Simcyp is, from early development on through mostly clinical development. And in particular, as we'll talk about, Simcyp Discovery is generally focused on some of the earlier aspects of drug development. So, an overview of Simcyp mechanistic modeling software. So, we develop. So essentially, we have three major functions. We've got quantitative systems pharmacology, which we have several platforms in that area, including immuno-oncology, immunogenicity, neurodegenerative, and COVID-19, as well as vaccines. And then we have physiologically based PK, which is the remit of the historically of the Simcyp platform. And then finally, we also have offerings in safety and toxicity. So PBPK is what the body does to the drug. So literally, it's like PK, pharmacokinetics, but with many, much more physiological sophistication. Quantitative systems pharmacology is what the drug does to the body, and generally, the implication is targeted, meaning the desired effect of the drug, whereas quantitative systems toxicology is more around the off-target activity of the drug, the things that we don't really want the drug to do, but we have some platforms around that. The Simcyp simulator is the leading technology for PBPK. It's used by 11 global regulatory agencies, and you essentially can be confident that you're using exactly the same tool as the regulators. It has supported the approval of almost 90 drugs, with more than 275 label claims using Simcyp in lieu of clinical studies. The majority of top 25 biopharmaceutical companies are members of our Simcyp consortium, which is comprised at present of 37 companies in total. And as mentioned here, quantitative systems pharmacology is rapidly advancing using both Simcyp's software and services. And our newest member of the family is the quantitative systems toxicology. So, where does, we use the PBPK across a pretty wide spectrum of the R&D cycle, including even not only discovery and non-clinical, but early development and late development, including in later development, we can help with drug-drug interactions, dosing recommendations for pediatric and special populations, organ impairment modeling, and as mentioned before, label claims in lieu of actual clinical studies. That's the wider Simcyp platform. Simcyp Discovery is generally targeted for applications earlier on in drug development, including the discovery phase and preclinical phase, and then also some early development. And one of the major applications of this new platform is to derive first-in-human dose estimates based on PBPK methodologies. As mentioned earlier, Simcyp, the overall simulation platform, has been used to make a 275 and growing number of label claims for 90 novel drugs across a variety of therapeutic areas. And this is essentially, a summary of these development programs that we were able to assist by helping make claims without actually having to do clinical studies. Okay, at this point, we are going to go ahead and play our pre-recorded demonstration of the Simcyp Discovery platform, after which I'll rejoin the discussion to discuss a few, like a wrap-up of the overview and next steps. And then, as I mentioned, after we will have a brief Q&A followed by Ian's detailed technical discussion. And thank you, Hannah, for that demonstration, pre-recorded though it may be. And reiterating on what Hannah said about the 4 major use cases, one of the major ones is estimation of first-in-human dose using PBPK approaches and incorporating potentially animal data and projections based on animal data. Again, this is not a by any means definitive, but it is a great facility for assessing whether or not a new compound has a high degree of DDI risk, and then based on that, you know, a more in-depth analysis can be performed. We also develop this to help prioritize and select compounds, and then with that in mind, there is a batch processing capability within Simcyp Discovery to facilitate processing of several different molecules with regard to projecting either first in human or some of the DDI as well. And then, of course, another aspect is formulation science, whereby based on in vitro characteristics, we can help to optimize the characteristics, be they physiochemical, as an example, of new compounds. So here are potential next steps if you are interested in Simcyp Discovery. For a single license, the price currently is $60K US dollars per year. If you're interested in evaluating the software, we can provide a 5-day evaluation license available on request. We will also provide technical support free of charge to all evaluators, which you can access through our support email address, which is simcyp.support@certara.com. If we see a lot of the same questions, we may schedule an office hour session for folks to come and answer questions and for us to potentially present tips and techniques. If you decide that you would like to evaluate the product, or simply due to your presentation, your participation today, we might be in touch with you to gauge your interest in the product and see if there's anything we can do to address any questions or concerns you may have. As a guideline, if you've used our first-in-human consulting services 2 or more times a year, essentially the cost of the license should pay for itself relative to the cost of a consulting services. So at this point, I would like to open up the the mic, if you will, or the chat facility with regard to questions. As I mentioned, if they're of a detailed technical nature, it might be better to hang on the line for Ian's and in case he already has answers for your questions in his presentation. Okay, not seeing any questions yet. I think we'll give maybe another minute or so, Ian, and then if we don't have any questions, then you can start with your presentation.
I have a few questions coming in. One moment.
Okay, great. Thanks, Trudy. Here we go. Ian, they'd like to, this question is about the batch mode. They'd like to hear more about, what capabilities are there in support of compound prioritization. So Ian or Hannah, maybe you could address that.
Yeah, so with the batch mode, it lets you queue up either workspaces or compound files for as many compounds as you're interested in, really. And then it will automatically just run through them in terms of making predictions of the human or whatever pharmacokinetics based on the input data you've given it, and it will produce all of the results for you, and then you can have a look at the predicted concentration profile for all of the compounds and see what the different compounds look like. So there's a way to just in an Excel sheet create all of the compound files. You can load that into the simulator, make the compound files, and then run them all automatically through batch.
So let's see. 1 second here, I'm having a little trouble. Let me resize the window. Make trouble reading all the questions here. Can you give some insight on how the software handles input data with respect to heterogeneous input data? I think they mean the structure. Yeah, I think what they're implying there, Ian, is like what sort of input formats does the software support?
Yeah, so I can go through that as we go through the presentation, but in terms of the data, it's normally numerical data for each of the parameters. So within the discovery project, it's not predicting things from structures, so there's no need to load structures into discovery. It's based on either predicted properties or available data like physicochemical properties, protein binding, etc. So I'll talk about that as we go through. But
yeah, and you either add it directly to the simulator or via the Excel sheet in batch mode.
So one of the other ones is, do you foresee it being useful to integrate this software with D360?
Yeah, that would be a very good approach, and that's something we've been discussing internally for a while now. But yeah, certainly you could pull all of the input parameters you need from D360 or from any other, I guess, data warehouse. Use those to populate the input sheet, create the compounds, and just run them through in batch. So in theory, you could do at least predictions based on available information for as many compounds as you wanted to. But yeah, that's certainly something that we've been discussing internally, that link between this product and D360.
Next question is the software, can it be used for large molecules such as monoclonal antibodies?
No, so the discovery is really focused on small molecules. Obviously, within the main Simcyp simulator, you can simulate large molecules and antibody drug conjugates, but the Simcyp discovery is purely focused on small molecule drugs.
Okay. Nice presentation. Is it possible to predict physiochemical properties from the structure of the molecule?
So it is possible to do that, but not within the Simcyp discovery. So Simcyp discovery is using like derived parameters, so it's not structure based.
Right.
So there are a few parameters we can predict within the simulator, but they require the phys-chem parameters, so we can predict protein binding and blood to plasma ratio, for example, and liposomal binding.
Yeah,
Sorry,
yeah. Use some
Hannah, I couldn't
examples.
hear that last part, actually. I heard other simulators. Yeah.
Sorry, you can predict some of the in vitro parameters within the simulator, so plasma protein binding, blood to plasma ratio, microsomal binding, etc. But they require physicochemical parameters.
Great, thank you. Is it possible to add preclinical species besides what's available?
Yes, possible to add other species as well. There's some of those, depending on what you're interested in, we already have some of the information, but yes, it's possible to do that.
Okay. For early discovery with transporter substrates, what is your suggested approach to address if the level of characterization JMAX or EC50 is routinely not available? That sounds like a question that might be better kept until after your presentation. Would you agree?
Yeah, that's fine. We can deal with it then.
Yeah. Which scaling method is used for human prediction is another one. Maybe that's another, that sounds like another technical one that maybe, you know, your presentation might speak to.
Yeah, that should be, well, that is addressed in the presentation, so that's fine.
Okay. Let's see. Does your software take individual subject data or average data only?
So you can make predictions for populations. So there is physiological variability included within the model for things like enzyme levels. So you can look at the effect of polymorphisms for polymorphic enzymes. So those kind of things are included. So you can make simulation results for populations, but it's not a full, they're not fully described populations, so there isn't variability on every parameter, but things like the absorption parameters, the enzyme levels, the variability there can be taken into account and can be used to see what, you know, what kind of impact that's going to have from person to person.
Thank you, Ian. We'll just have a couple more, and then I think we'll launch into your presentation. Will Simcyp Discovery be replacing the existing Simcyp Animal Simulator, or will this Simcyp Animal continue to be offered as a standalone?
So at the moment, the plan is to offer both. So it's not a direct replacement, although some of the features obviously are similar.
Yeah. So the next one, and maybe this is another one that we can hold off on until after your presentation, is could you tell what is the difference between non-compartmental analysis and one-compartmental model? That I think your talk might speak to some of that. Is that right?
Yeah, we'll cover that as well.
Great.
Answer that at the end.
And I'm going to try and answer this one on my own, and then you tell me if I, if I got it right or not. It says, I saw Simcyp supports IV and oral administration. How about inhalation, intra-articular, or local administration? And I think the answer to that is Simcyp Discovery only handles IV and oral. Some other routes of administration can be, essentially handled in the the full Simcyp simulator. Is that correct?
Yeah, so in this one, we don't have inhalation or dermal, but we do have inhalation and dermal models in the full Simcyp Discovery. In the full Simcyp simulator.
Great. All right, so I think let's pause there, and then Ian, if you could go ahead and walk us through your presentation on the technical details underlying the platform.
Yeah, sure. So hopefully you can see the slides okay and can hear me okay. So what I want to do in this presentation was just kind of give a brief overview of what we're doing within the Simcyp Discovery Simulator. So as you know, for physiologically based models, what we're doing is integrating physiological information, so information about organ volumes and blood flows, information about weights of different tissues together with drug-specific information, and then trying to make predictions of the absorption, clearance, and distribution of the compound so that we can predict the concentration in plasma and in different tissues. And so really what we were thinking with putting together the discovery simulator and launching it was really focusing kind of on the transition from preclinical to clinical. So trying to make the most of all of the information that's available at that stage in terms of all of the human in vitro data, any preclinical pharmacokinetics. So take everything we know about the compound, put it into the PBPK model, and then make predictions about what's going to happen when we dose the compound to humans. And so, as Mark alluded to in his presentation, what we've done within the simulator is to make available physiology libraries for mouse, rat, dog, monkey, and human. And as I mentioned, you can customize these populations. So if you're interested in other species, it's possible to add those in as well. So just a quick overview. Really what we've done then is to combine elements of the Simcyp simulator and Simcyp Animal into a new tool. So we still provide populations where we have demographic data, information about liver size and some of the liver parameters we need, tissue volume, flow rates, and also the brain. The compound information is simplified, so we need some information about the physicochemical and binding properties of the compound, and we want then to make predictions about the absorption, distribution, elimination, transport, and pharmacodynamics. And I'll explain as we go through the slides how we do that. As we mentioned in the questions, we can support batch screening, so we can run libraries of compounds through the tool. And obviously, as we discussed, it's possible to think about linking up the discovery tool to things like D360. Really, what we're aiming to do with this tool is really think about predicting first-in-human dose using physiologically based pharmacokinetics. Generally, as Hannah showed in her presentation, we're using verification stack on preclinical species, although that's not always required. And obviously, we want to provide early pharmacodynamic readouts based on the pharmacokinetics, target concentration, drug potency, because that will drive the dose, and really that's what we're interested in setting for the first-in-human studies. We have included non-compartmental analysis and compartmental fitting to help parameterize the model. So really, if you're only interested in looking at the absorption, you can use those tools. And also, as I'll show, and Hannah showed in the demonstration, there's a DDI liability tool as well. So if we think about what we're trying to do when we're predicting the oral pharmacokinetics of a compound, so what we want to have some idea of is the oral bioavailability. And to get to the bioavailability, we need to be able to predict the fraction absorbed, the fraction escaping metabolism in the intestine, and the fraction escaping first-pass metabolism in the liver. And so in this presentation, I'll show you what tools we have available to make predictions for each of those parameters. And if we're thinking about the maximum concentration, that will depend not only on the bioavailability of the compound, but also on the rate of absorption and the volume of distribution. And again, I'll show you how we can predict the rate of absorption, volume of distribution within the Simcyp discovery tool. In terms of data requirements, so this is what we need to have available to be able to put together a PBPK model. So we need basic physicochemical information, so molecular weight, log P, pKa. We need plasma protein binding, blood plasma ratio data. So here we're looking in vitro binding data, so for humans or for other species. We need some in vitro intrinsic clearance data, so metabolism data, and I'll talk about that a little bit more in a few slides' time. We need an estimate of passive permeability, so that can come from in vitro systems like MDCK or CACO. It can also come from physicochemical properties. If we're going to go to quite detailed modeling of absorption, then we need information about solubility. And often that will be measured in biorelevant media. We need an idea of what the target exposure, the dosing regime, and what kind of formulation we're going to use in human. And so obviously within the simulator, we can look at different options here and see which ones will give us the desired profile. If we're comparing to known data in preclinical species, we can use in vivo data for model verification. Sometimes that's useful to help show that the models are working well, or what adjustments we need to make. In the animal species, we can then think about whether those are relevant for the human situation. And obviously, if we want to use the static DDI calculation, we need some inhibition parameters. As Hannah mentioned in the questions, there are some prediction tools available. So we have prediction tools from physicochemical properties for fraction unbound in plasma and microsomes, blood plasma ratio. We can also make predictions of solubility, permeability. So again, they can be quite useful early on. And obviously, as you get more information, you can refine the predictions and then update the models. We also have tools to allow allometric prediction from species to species, or from preclinical species to human. So all of those are available. If I just walk through the different approaches we can use for absorption, distribution, metabolism, and excretion. So firstly, thinking about absorption. Within the discovery simulator, there are two models available. So first one's a fairly simple first order absorption model. So here we're taking some information, so physiology information, things like the radius of the small intestine in the different species, and an estimate of the permeability. So really, the main thing that we need to be able to predict is the permeability of the compound. If we know the permeability of the compound, or we can predict it, together with the physiology information, we can predict the rate and extent of absorption. We also have kind of more advanced absorption models available, so the ADAM model, where the intestines split up into 9 different compartments. And within this model, then within each compartment, we can think about dissolution of the compound, absorption of the dissolved drug, precipitation, and once the drug's absorbed, it will pass into the intestine. Within the intestine tissue, there can be metabolism. It will pass into the portal vein through the liver, and then into the rest of the body. So those are the two main absorption models available. In terms of predicting permeability, there's a number of different options available to you. So you can make predictions based purely from physicochemistry, just using the permeability prediction. You can use in vitro data from CACO2 or from other cell lines. You can also calibrate the permeability of your compound against reference compounds and use that as an input. And you can also define your own model for any system that you're interested in using. So there's quite a lot of flexibility in the ways to predict permeability. I guess the most often ones are to use the physicochemical data and the mechanistic permeability, or to use data from in vitro. Within the intestine, you can also account for metabolism. So there are different ways that this can be done. So we can measure unbound intrinsic clearance. And so we can do that either within things like intestinal microsomes or intestinal slices, and use those with physiological information, like microsomal protein per intestine, in a kind of direct scaling approach. Also, we can take information from liver microsomes where we know the relative enzyme abundance in the liver and the intestine, and use those to scale. So all of those allow us to get an estimate of the unbound intrinsic clearance in the whole intestine for the species that we're interested in. In terms of calculating FGE, so the fraction, or the fraction of compound that's not metabolized on first pass through the intestine. For the first order model, we do that using this Q-gut model, where FGE depends on Q-gut and the unbound intrinsic clearance in the whole intestine. This Q-gut depends on two things. So it depends on the physiological data, so it depends on the villus blood flow and the interest in the species we're interested in, and also the permeability of the compound, which again is directly related to the permeability in the species that we're interested in. When we have high FGE, so values close to 1, then we see that with compounds with high permeability, low unbound intrinsic clearance in the gut. If we have low permeability and high metabolism, then FGE would tend towards 0, and you end up with a lot of metabolism occurring in the intestine. So for the first order model, that's the approach we use. And the Adam model is a little bit more complex, but we calculate the FGE dynamically using information about the intrinsic clearance in each of the segments of the permeability, and the enzyme concentration of the compound. But basically, both models will allow you to get an idea of how much the intestine is contributing to first pass elimination of the. If you use the Adam model, you can look at different formulations. So you have a number of different options available to you. So the simplest one is solution, where the compound's in solution at all times. But using the Adam model with solution still allows you to look at regional differences in metabolism and transports. It can still be a useful way to go. The compound can be dosed in solution, but depending on its solubility and its properties, it can precipitate to fine particles. We can also look at dosing suspension, so where the compound's dosed as a mixture of pre-dissolved compound and particles. And we can also account for a number of different solid formulations, including immediate release tablets. Immediate release, we can look at using dissolution profiles, where we're looking at dissolution of the compound. So in that case, we're not considering precipitation, but we can also look at release of the particles from the tablet. And then once the particles are available, then they're handled through the diffusion layer model. So they go into solution, and depending on the properties of the different compound in the different individuals, you may or may not see precipitation. You can account for enteric coated formulations, either granules or tablets, and we can also account for modified or controlled. Within the Adam model, there's quite a lot of options available to investigate different formulations and to see what the impact will be on the pharmacokinetics. There are other tools that we have available for helping to parameterize some of these models. And so you can look at modeling dissolution rate profile and parameterizing the diffusion layer model in the Simcyp in vitro. I'm not going to go into that in any more detail here, but if you want more information about it. So just to summarize, in terms of absorption, we can predict the fraction absorbed and the rate of absorption using either the first order or the Adam model. Both models allow us to look at the metabolism in the intestine, but the ADAM model allows us to look at formulation, food effects, and also allows us to look at the effect of transporters in the intestine as well. So there's a bit more flexibility with the ADAM model for looking at extra. Moving on to quickly go through prediction of distribution. So for predicting distribution, we have a minimal physiologically based pharmacokinetic model where we define the liver and the gastrointestinal tract as mechanistic, fully parameterized organs, and the rest of the body is lumped into systemic compartment, and also to handle kind of compounds where there's biphasic kinetics, you can also use single adjusting compartment to account for biphasic disposition. This is a bit of a kind of middle out approach to, I guess, for the minimal PBPK. So you can predict the overall volume of distribution using estimates of the tissue volume and the partition into each of the tissues. So that's done from physicochemical properties, and in a couple of slides time I'll explain the different methods we have available to do that. And then the volume of the systemic compartment, VSIS, then depends on the estimate or the value you use for the volume of distribution at steady state, the physical volume of the liver, and then any volume that you have for the single adjusting compartment. Within the Simcyp discovery tool, we've also implemented compartmental models, so you can just model the systemic compartment as either 1, 2, or 3 compartments with the rate of transfer between them defined. And the idea with this compartmental model is that you can just fit the IV profile if you have it in the particular species you're interested in, and then use the model to simply look at what's going on within the intestine for looking at the absorption. So that's really what the main use of this this model. And then obviously we have the full PBPK model available. So here we define all of the different organs. The definitions are anatomically correct in terms of the connection with blood flow and the size of the different organs. So to parameterize the full PBPK model, what we need to be able to predict is the partition of the compound between the tissue and plasma. That's shown in the equation there as the P-TP ratio or KP ratio that used interchangeably. And we have 3 methods available for calculating or predicting that available within the discovery simulator. This is really, I would say, the most frequently used model for predicting the distribution of. So the 3 methods that we have available. So the first one's based on the original Poulin and Tile papers, as modified by Bereskovsky in 2004. So here we're looking at movement of drug from the plasma into the tissue, and then binding of the drug within the tissue to neutral lipids and neutral phosphate. The second model we have is based on the work of Judy Rogers, Malcolm Rowland, where they split the tissue into the intracellular and extracellular water, and then within the tissue we're looking at partition of the compound into not just neutral lipids and phospholipids, but for basic compounds, also the interaction with acidic phosphate. And the last method is one that we developed within Simcyp, so it's based on the approach outlined by Rogers and Rowland, but also considers movement of charged species from the extracellular water into the intracellular water. And so that movement depends on the charge of the compound, permeability of the compound, but also on the membrane potential. And so all three of these methods are available for you to use. There's a bit more increasing complexity as you go from left to right, but really the input data is the same. So you're using information about the physicochemical compound to prediction. Usually, for a compound, you would try all three methods and then see which ones are given the clinical species and use that for the human situation. So just some notes on the KP method. So there are some common assumptions between all three methods. Let's say the biggest difference between method 1, method 2, and 3 is that method 2 and 3 consider compounds differently, whether they're acids, bases, or neutrals. And method 3 allows the ionized fraction to be permeable and to distribute into the tissue. Okay, so briefly to talk about prediction of clearance. So the approach that we've adopted within Simcyp and also using discovery is based on in vitro, in vivo extrapolation. So typically, this would be based on data in microsomes or hepatocytes from the species that you're interested in. So look at the metabolism in terms of either parent depletion over time, as shown here, or formation of metabolites. Both of those can be incorporated into simulations. So from the in vitro data, we can work out the intrinsic clearance. We can correct that for binding in the in vitro system, so from microsomes or hepatocytes. And then we can take the in vitro unbound intrinsic clearance, scale it up to an unbound intrinsic clearance per gram of liver, or once we know the unbound intrinsic clearance per gram of liver with the liver weight, we can scale up to the. This is a general kind of approach that we use for scaling metabolism data. As it says here, you can also make predictions from within the PBPK model, we're assuming well-stirred conditions, and so this is just showing the equations for the well-stirred model. I guess one of the main things we're assuming with the well-stirred model is that the it's unbound within the tissue is equivalent concentration unbound transporters. So we can also switch to a permeability limited model and look at the effective uptake in efflux transporters in the liver as well if we want to. There's a fair amount of flexibility for different inputs, and they're just shown on this slide. You can use microsomes, hepatocytes, or recombinant enzymes. You can use intrinsic clearance. You can use VMAT if you have metabolite formation data. So there's quite a lot of different flexibility. In terms of excretion, you can account also for renal clearance. So we can include renal clearance within the compound files, and also biliary clearance as well can be included. So again, with the permeability limited models, we can include that by specific transporters, or we can just include biliary clearance as a both of those are possible within the discovery simulator. So if we think about how successful PBPK predictions are, there's quite a lot of literature out there. So this is just summarizing some of it. So this is looking in particular at preclinical studies. So generally, volume predictions are within twofold. There can be under prediction of clearance, and I'll talk about this a bit more when we talk about the human clearance prediction. But commonly now, people account for any known systematic under prediction within in vitro systems. So if you take that approach, you can get usually reasonably good predictions using in vitro in vivo extrapolation. And there are a couple of publications highlighted here. So one from Roche, one from J&J, where they looked at prediction of tissue levels in preclinical species and showed that they could get reasonable predictions using this kind of PBPK approach. A little bit later, also, some work that Hannah did showed that using PBPK for early toxicology studies was more accurate than kind of linear scaling of low doses. So again, another kind of application of. In terms of predictions in humans, so there are a number of studies showing that the AUC, Tmax, Cmax, bioavailability clearance, VSS, normally predicted within twofold for more than 50% of compounds that have been investigated. And this was some work that Hannah and I did when we were at Pfizer. So looking at PBPK versus single species scaling. And actually for the data set that we looked at where we had intravenous data, the clearance predictions from pretty well compared to single species. If you look through the literature, there are a number of publications, as I mentioned, where they tend to see an under prediction using in vitro, in vivo extrapolation. And so generally, to get the most accurate predictions, most people now use an empirical scaling factors, so they can be determined, and obviously. And just a couple of publications highlighting this. So this one from in Houston, so this was published in 2012. When they looked at a number of different PBPK or different scaling approaches to predict clearance, and showed that from in vitro data, if you correct for the bias, then you. And more recent paper, again from the group in Manchester, where they looked at scaling factors and used different scaling factors depending on the system and the prediction, and again showed that leads to reasonably successful predictions. And this is something that we've routinely used in consultancy projects. Just to show you a couple of kind of applications. So this is a publication done at Pfizer, so a few years ago now, but looking at using Simcyp in discovery. And so what they were doing was trying to select between different compounds based on the window between where they thought the efficacious concentration was and the concentration where they were expecting to see adverse events. In this case, the adverse events were driven by Cmax. So they used a first order model, minimal PBPK. So they had physicochemical data, in vitro clearance, and obviously information about the binding of the compound in the in vitro system. So this is kind of what they were doing for different compounds. So you can see the predicted concentration profile shown by the black line. The dashed lines were population estimates for the fifth and 90%. And you can see that for the first compound, there's reasonably space between the concentrations, maximum concentrations that would give you concentrations above where you would expect to see efficacy and where you would start to see adverse events. For the second compound, there's still a reasonable gap between the concentrations where you would see efficacy and the adverse events starting to occur. and for the third compound, in this case, window between where you would see the efficacious effect. So again, just showing an application where making predictions for compounds early on, you can kind of use that information within your decision making. So just some general guidance. So generally, when we're doing the consultancy type projects, we do pre-verification in preclinical species to kind of build confidence in the data we have and the assumptions we're making, and then to make similar assumptions in human. As I mentioned, clearance can be underpredicted from in vitro systems, so generally correcting for that's necessary, and that's generally the approach that we take. We often explore a range of different clearance prediction methodologies. I think in general, the volume distribution is better predicted across species. And so we also look at the tissue composition equations against any kind of in vivo data that's available and see how well the different models work. We generally predict impermeability, like I said, either from CACO or MDCK data or from mechanistic permeability model. And usually if the compound has low solubility, then we'd also take that into account. So use a more complex model like the ADAM model and factor in what we know about solubility in biorelevant media. Obviously, the regulators are starting to put emphasis on modeling in simulation in starting dose calculations. So this is typical kind of workflow, I guess. So you have some AL from a toxicology study, you make an estimate of human exposure, you can select your most sensitive preclinical species, account for differences, and then predict the human equivalent dose. Within the EMA guidance, there's a bit more of an emphasis on PD effects. So really thinking about the pharmacodynamics preclinically and then what you expect in the clinic, use that to help decide the safety factors you need and the effective human dose. So to help with this, what we've done within the Simcyp discovery is we've made a number of pharmacodynamic models available. You can connect the PBPK model concentrations in any of the compartments to a pharmacodynamic effect. So we have the kind of standard direct response models, so linear Emax, sigmoid Emax, and power models, their own direct response models, and also custom models as well. So there's quite a lot of flexibility in custom models. You can basically do any kind of pharmacodynamic model that you want, and we can help with those if you need help to set. And the last thing that we did with the discovery tool was to include static DDI approaches. So based on the regulations from FDA, EMA, and PMDA, and as Hannah showed within the demonstration, that you can look at basic cutoff models or the more mechanistic static models within the discovery. I think that's the last slide. So I'm happy to take any questions. And
thanks, Ian. Let me check the Q&A window here. Or let's see. The question is for the DDI predictions. It looks like a simple yes/no answer. Does it incorporate both metabolic and transport potential for interaction?
Yeah, so you can look at the transporter interactions as well, but still based on the static guidelines or the static models within the guidelines. But yeah, both are there.
just giving a few more minutes for other questions to come through.
Yeah, so there was a question about transporters. So you can account for transport in the intestine and in the liver within the model, so uptake and efflux, and obviously also within the brain. So it is possible to look at the effect of transporters in those 3 organs as well with the discovery tool.
And I think also going back to the question around the different input parameters and being able to accept a diversity of assays and things, you'll have seen from Ian's presentation that for many of the processes we can account for multiple types of input data. So for example, with absorption, CACA2 versus MBCK, versus being able to predict it from his chem properties, etc.
Okay, Danielle, are there any other questions coming through? Or Trudy?
No other questions at this time.
Okay. Well, we want to thank everyone for your participation today. As mentioned before, we'll be in touch to get a better understanding of your level of interest and/or if you have any questions or other things we can highlight regarding the Simcyp discovery platform. And many thanks to our organizer and producer Danielle Pillsbury and Trudy Curtis, and then to Ian and Hannah for your presentations and input today.
Okay, thanks. I mean, if you think of any questions afterwards, then feel free to get in touch and we can answer those. That's not a problem. So thank you.
Yeah, thanks very much, everyone.
Okay,
Bye.
thank you. Thank you, everyone. This concludes today's webcast, and I want to thank everyone for attending.